When people think of inheritance, they think of DNA being passed on from both parents. What people might not know is that there are specific DNAs passed on exclusively from mom or dad.
To put it into perspective, human cells have about 20 to 25,000 genes in every nucleus, and all that DNA is packaged into chromosomes. If that sounds familiar, it's because all of our genetic material is wrapped into 23 pairs of chromosomes, or 46 individual ones. And as diploid organisms, our mother and father are each equally responsible for half. And they grant you traits for how you look in the color of your hair or eyes, maybe the clefts of your chin, or if you sport attached or free earlobes. And it also accounts for those behaviors of how you respond to certain illnesses, the types of music you might appreciate, and whether cilantro tastes like soap. And also much more. Figured out by the genetic experiments first pioneered by Gregor Mendel.
In contrast, mitochondrial DNA is maternally inherited, and the non-recombining portion of the Y chromosome, which is paternally inherited, is slower to acquire mutations. Mitochondria is a small organelle with a big potential. They are the mitochondria, the powerhouse of the cell. They weren't always associated within a cell, though. They were previously their own standalone organism, a bacterium. It is speculated that primitive eukaryotic cells would have eaten them for food. But the early mitochondria were particularly good at making chemical energy, and this was just enough to cut a deal with the early eukaryotic cells to persuade them from digesting the engulfed mitochondria. Over time, they became symbionts with eukaryotic cells by losing much of their bacteria-like characteristics and DNA, only holding on to a total of 37 genes, of which 13 genes allow the mitochondria to keep doing what they do best, cranking out energy.
Mitochondria have another important characteristic. Their genes can be passed down through generations. Now, how do these genes move down the line? The answer might not be what you had in mind. Mitochondrial DNA are maternally inherited through non-Mendelian inheritance, whereas the nuclear DNA is inherited from all ancestors. Mitochondrial DNA has hundreds of copies, known as high copy numbers, while nuclear DNA has 2 copies. This is why mitochondrial DNA are easily found in bones, teeth, hair shaft, and aged fingernails. This makes mitochondrial DNA more sensitive for detection in samples. That's why mitochondrial DNA is a useful tool for forensics. It is used to identify missing people, criminals, and ID remains after natural disasters. So how does forensics do this? They use a technique called mitochondria sequencing to discriminate between closely related maternal lineages. This helps identify sequences closely related to the person and to highlight the variation among unrelated individuals. These unique features of mitochondrial DNA allows for tracking of individuals through generations and to contribute to how forensic lets us match unknown samples to family members.
So now that we're familiar with mitochondrial DNA being exclusively from our mothers, how exactly do our fathers contribute? Well, remember the Y chromosome? It's what's known as a sex chromosome, and only one copy of the Y is necessary to order male anatomy during embryonic development. Because males classically have an XY makeup, the Y chromosome is passed exclusively from father to son and nowhere else. But the Y chromosome is also considered a gene desert, as in there isn't much coded on it. So why is this so important? Well, in addition to the genes on it, making it responsible for sending out male structures, there are also genes that are responsible for making sure that they work. In particular, the directions for proper testes development, followed by sperm production, are also specified on the Y chromosome. And to keep it exclusive, the Y duplicates on itself and cannot recombine with the X chromosome, meaning that the sons inherit the entire structure intact from their fathers, making the Y chromosome an attractive map to track genetic forefathers and possibly pioneering migrators.
Speaking of pioneering migrators, have you ever heard of a guy named Genghis Khan? He's only one of the most feared conquerors of all time who established one of the largest land empires in history, a territory that spanned most of Central Asia and China. And did you know his Y chromosome is still present in roughly 16 million men? How is this possible? And how do we know this to be true? Using a technique called genetic barcoding, researchers have shown that Genghis and his descendants migrated even farther to distant places such as Eastern Europe and Southwest Asia, and their travels overlapped with many additional populations. But what exactly is genetic barcoding? Well, with recent advancements in DNA sequencing technology, scientists are able to compile information from the maternally inherited mitochondrial DNA and combine it with the information from the paternally inherited portion of the Y chromosome to generate records of human evolution. Think of it this way. Just like how a retail store uses barcodes to track items in inventory, scientists can use DNA barcodes to track events that result in an increase in genetic diversity. By tracing processes such as drift and migration of ancient populations, as well as revealing where early colonization events by pioneering migrators overlap, researchers can decipher trends in population genetics.
So you see, the mighty mitochondria are more than just the powerhouse of the cell. They can be used in forensic science to analyze maternal relatives.
But don't forget about the paternal relatives, responsible for the Y chromosomal DNA exclusively handed down from father to son.
And together, both mitochondrial DNA and the Y chromosome have the potential to help us build a better understanding of the accumulation of genetic diversity.